Hello, everyone. Welcome to Digest podcast. In this episode, we'll talk about biosensors and the importance of biosensors and some crucial fundamental papers in the field. Let me introduce you with our guest today. I'm happy to have Dr. Jagotamoy Das with me today. Dr. Das is currently a research associate professor at Northwestern University in the United States. Dr. Das, welcome to this podcast.
Thank you for having me here.
Let's start our discussion with your one of the most significant discovery. Here, you published a uh, crucial article, a groundbreaking article in 2021 in Nature Chemistry Journal. You developed a sensing mechanism in this article called molecular pendulum, which is a reagentless sensor that can detect molecular analytes in biological fluids. So, tell us about the importance of reagentless sensors and um, what was kind of the limitation in the field when you started this project, and how does this molecular pendulum work?
Well, that's a very nice question. The thing is, let me first discuss what is reagentless sensor. So, reagentless biosensor can monitor a target analyte in a complex biological mixture without addition of anything from outside. For example, you don't need to mix anything from outside, any reagents or anything. It just itself just all in one sensor that can give signal or detect uh, biomolecules that is related to different types of diseases.
Yeah.
So this type of sensors are very important for continuous monitoring or wearable monitoring. For example, if you see that glucose, personal glucose monitor or continuous glucose monitor that you can put the patch on the arm. It
Mm,
continuously detect glucose levels for that is useful for manage diabetes.
yeah.
If you think that
So
the
this method is
okay,
also called a non-invasive method. Is that right?
yes, we can say non-invasive method or minimal invasive method
Mhm.
because
Okay.
you're putting at least a needle or micro needle there.
Mm.
So we prefer to call this minimal invasion.
Okay.
Um, yeah, that's a pretty nice thing. Now, if you think the development of modern biosensors is like more than 65 years, and we have glucose sensors now that continuously monitor glucose. Um, but we do not have sensor that can continuously monitor proteins or other important biomarkers. This glucose is a very small molecule, right? And using enzyme, it is detectable. But for protein sensor, normally what we have, um, standard method is electrochemical ELISA. So you use enzyme or either electrochemical or optical ELISA. You use enzymatic reactions that give some signal or light that you can measure to detect biomolecules. Now
Okay.
the thing is for wearable sensors or implantable sensor, what you put in the body, you cannot add reagents from outside.
Mhm. Yeah.
So the sensor should be reagent as glucose sensor.
Mhm.
So that's why for wearable detections or implantable detections, this reagentless sensor is very important. Now the question is, reagentless sensor is crucial for this type of detections. There are some reagentless sensors for electrochemical sensors available. Say, for example, DNA scaffold-based sensors. There's pretty nice sensors, or aptamer-based sensors, pretty nice. But DNA scaffold-based sensor cannot detect antigen-antibody interaction. Antibody is important receptor for biodetection.
Yeah, yeah.
So we're thinking to develop something new that can be like adjustable to any types of receptors to detect biomolecules.
Mm,
So we came out idea we call molecular pendulum. So that use like if you know the constructions of biosensors, we have electrode, we use some biomolecules, we use
mm,
receptors here, maybe antibody or other things, maybe aptamers. Then it binds to like its target molecule. And
mm,
for like getting signal, people use that I mentioned enzymes or maybe nanoparticles or other things. But for wearable case, we can use like anything reagents from our side.
yeah.
So we built a sensor. We developed two probes, modified two DNA probes, the surface. One probe contains one redox molecule. We use ferrocin that gives electrochemical signal,
Yeah.
and another bound to an antibody that binds
Mm,
to its target molecules. So this strand with antibody, when you apply like positive charge at the electrode surface, so this is negative charge DNA.
yeah.
So positive charge attracts this DNA like that, right?
Mm-hmm.
So it behaves like a molecular pendulum when you
Oh,
apply
okay.
positive charge. Now the thing is, this molecular, this pendulum is a ball. If this is small, it falls faster. If it's big, it falls like slowly.
Okay.
So now this two stage we call like bound stage or unbound stage. When antibody binds to its target protein, is bound, and when it doesn't bind to its target protein, is unbound. So unbound state is smaller; it falls quickly, and when it binds to target protein, which is now bigger, it falls slowly. So this time lag that we can translate using one electrochemical technique, we call this chronoamperometry. So this kinetics of the systems can be translated to measure current using chronoamperometry, one electrochemical
Mm,
technique. This we can detect like target molecules. As now, this is like antibody-based sensors. You can use any antibody to detect its target. So it's like universal for all types of target proteins. You can. We also develop like changing antibody with aptamer to detect uh, disease markers related to heart failure. And we
mm,
tested the sensors in all types of biofluids like blood, urine, saliva. It works for all of those. We tested many different proteins biomarkers related to different diseases. For example, heart failure, cancer, allergy, and many others. We tested placing like a small probe or electrode um, in oral cavity of a mouse, which is induced for heart failure to detect. It can detect like heart failure markers from live animals continuously, and the sensor pretty well detected that.
so every time I have a just small questions. Every time, depending on the purpose, you need to change the antibody inside the sensor, right?
That's correct.
Mm.
Or
Okay.
you could like develop like multiplex sensors.
Mm.
Um, like on one chip, you could like plate multiple electrode. Each
Right.
of the electrodes can be used for like one
Oh,
biomarkers.
got it. That's wonderful. Yeah. Multi-purpose
Multiple.
device. Okay. So here's another question. So as this is an antibody-based um, uh, system, so do you think it is ultimately if you make a device, it will be temperature sensitive or there will be issue with storage and those things?
Yeah, storage and stability is very important things.
Mm,
So there's like many things could be involved there. High temperatures could change the surface. But for ambient temperatures like body temperatures or room temperature, this is quite fine. But still, for like long time continuous monitoring is like challenge. We are working on that.
Yeah, yeah, right. Because sometimes for diagnostics purpose, people develop RDT or rapid diagnostic kits that you based on some gold nanoparticle and those things. So uh, sometimes those are helpful, but they are just one-time use. You cannot monitor continuously, so that's a limitation.
That's correct. So for we using different techniques, we are able to like use the sensor for in vivo monitoring like almost for 7 days. We are continuously working to like improve the sensor stability for longer time measurement.
Okay, okay, thank you. That's a wonderful device, and I think it will help future researchers and also future and diagnosis purposes. You have so many wonderful publications, and we may not be able to discuss all, but let me show one of the first publication, crucial publication you have, and we can talk a little bit about that and what happened after that. So here's in 2006, you published a JAX article where you developed a nanocatalyst based electrochemical assay for proteins. So, uh, can you please tell us a little bit more about that? What you did there and how did it unfold since then?
Well, so the thing is that stability of the sensor that we already talked, right? So, and I talked about like ELISA-based sensors that
Mhm.
you have uh, antibody binds to a target molecule, then you use secondary antibody with like enzyme and use enzymatic reactions to like detect the target molecules. So this is like a very standard gold method using for biosensors. But those sensors like for one-time use.
Yeah.
And definitely like enzymes are very expensive and not very stable.
Yeah.
Um, so I thought if I can replace the enzyme with something more stable, and then that gives like similar catalytic reaction. So I use gold nanoparticles catalytic property. I'd like to say here, like I was one of the pioneer who used like catalytic activity of gold nanoparticles to develop the biosensor.
Mm.
So in that case, it's like pretty simple, very similar to ELISA-based sensors. So I
Mhm.
have primary antibody that binds to its target molecules. I have secondary antibody that already conjugated with gold nanoparticles. And gold nanoparticles convert one electrochemically inactive molecules to electrochemical active molecules that produces signal. So now the signals change is directly proportional or depends to like target concentrations. So we're able to like detect prostate specific antigen. This is like a marker for prostate cancer, and that gives like a very good sensitivity. We detected very low amount of PSA for like detections. So it was pretty nice paper. Well, like we submitted the paper. It's like very interesting. Few of the reviewers accepted the paper without any change, just with some minor comments.
Okay,
Uh, a
that's very
paper
rare.
like in Jax, like published from the reviewers comment like that is like I felt like so proud about that.
Yeah, that's wonderful. And after you moved to Canada, you had another wonderful research where you developed an ultra-sensitive universal detector based on neutralizer displacement. Could you please explain a little briefly about this one? what you did here and what was kind of the update?
Yeah, of course. Um, so I talked about Elijah-based sensor. I talked about nanoparticle-based sensors.
Mm-hmm.
So those are like we call sandwich-based sensors. So you have like antibody, you have target, and another antibody that sandwich target. So you need like
Mm-hmm.
additional reagents that you need to add for sensing. Uh,
Yeah.
so another simpler way to detect nucleic acid, if you had like a probe that is specific to the target nucleic acid, and say probe is neutral, and you use like we use for that purpose we call hexamine ruthenium, that is positively charged complex of ruthenium that electrostatically attracts to negative charge. So when target binds to the probe, so the surface is now more negative due to negative phosphate backbone. It
Mm-hmm.
it binds to hexamine ruthenium
Okay.
electrostatically, and it produces signal when you reduce ruthenium 3 to ruthenium 2. That tells like uh, how many target molecules binds to the like probe. That's how we
Mm-hmm.
can detect like target molecules. So now this system, if you think you have probe, you have nucleic acid, you can detect that. But for this simple, nice system, you cannot detect like um, small molecules or target proteins that doesn't have significant charge,
Yeah.
right? Because this is charge-based sensor.
Right.
So I thought, okay, if I use something that can neutralize charge of the DNA probe,
Mm-hmm.
and after target binding, if that removed from the surface, so charge of the DNA probe will restore. We can use same system for detections of many different target molecules.
Okay.
So we call this neutralizer displacement assay. So for example, say I have a DNA probe here. I hybridized neutralizer that is positive charge and DNA backbones that can hybridize to that, and positive charge neutralizes that negative charge of the DNA.
Mm-hmm.
And when target molecules comes, it's uh, it kicks out neutralizer, so negative charge register from the like probe, and we
Mm-hmm.
can use like that ruthenium and ferricyanide system. Ferricyanide amplify the current from ruthenium chemically system to detect the target molecules. Now,
Yeah.
if this is target is DNA or nucleic acid, we can detect that obviously. So for detections of protein molecules, we use DNA aptamer, we use neutralizer, then target molecule binds to the aptamer, it's folded, and due to that, it releases neutralizer.
Okay.
So we can use ruthenium ferrous system to detect that protein.
Mm-hmm.
Similar for small molecules, say we tested cocaine and ATP. Um, when like it binds to a aptamer, neutralizer release, and we can use that electrochemical system, hexamen ruthenium and ferricyanide to measure the electrochemical current. So
Mm-hmm.
that sensor can be used to detect nucleic acid, protein molecules, small molecules. So we call this universal sensor because we don't need to like depend on the charge of the molecules.
Mm-hmm.
Such as for the other assay, we had to like depend on charge of the nucleic acid. But now we have freedom. We can use whatever molecule says either charge or not, using this
Mm-hmm.
neutralizer displacement method.
Okay. Okay. So, can you give me an example in real life where can we use this method practically? Let's say to for diagnosis purpose or to design a device, wearable device or something. So can we use this method for that?
Uh, No, you have to use this method for bench stop study, not
Okay.
like
For research use.
yeah. Um, because you need to add like hexamine, ruthenium, and ferricyanide in the system,
Mhm.
right? So this is not reagentless.
Okay.
It's simpler than ELIJA, way simpler,
Mm-hmm.
but it's still not reagentless. We have
Yeah.
to use some reagent to get electrochemical signal. So
Mm-hmm.
for variable purpose, we cannot use that beautiful system. But for like other sensing cases on benchtop, or like ex vivo, um, or like we can use that for
Okay.
detection of all types of nucleic acid biomarkers, protein biomarkers. We tested that system that time to detect bacteria, bacterial RNA. We tested some other protein biomarkers. We tested ATP
Mm-hmm.
small molecule. We used this system to detect cocaine, that is the drugs of abuse.
Mm-hmm.
Um, yeah, so for many different types, we can use that not in vivo, but ex vivo.
Okay, got it. Uh, that's really helpful. Okay, let me pull up another project that you worked on, and you made a really important publication from that, which is this paper published in JAX in 2016, where you developed a DNA clutch probes for detecting circulating tumor DNA. So, what was the story behind this article, and what did you do there?
So the story, we wanted to develop assay that can detect cancer-related
Hmm.
biomarkers.
Right.
Now, for cancer-related biomarkers, it's better to like target something that is quite or closely related to cancer,
Hmm.
not all types of nucleic acid. So we targeted mutated nucleic acid. So mutated nucleic acids are directly related to cancer. But detections of mutated nucleic acid, DNA or RNA, are very challenging because mutations means, say, one of the DNA bases changed to another one from the wild type. So their concentration in blood or serum is very low
Hmm.
compared to wild type. So when you have like probe molecules there, and you have a lot of wild type molecules, DNA molecules or RNA molecules, only few mutant molecules. So it's hard to distinguish that mutated nucleic acid from wild type because wild type's amount is way, way higher than mutated molecules.
Yeah. Hmm.
So we came out with an idea if we could like block wild type nucleic acid, keeping only mutated nucleic acid available that can bind to
Hmm.
um, probes.
So, by wild type, you mean the regular nucleic acids that are present in blood and in
Correct.
biological fluids?
Correct.
Okay.
So we developed electrochemical clamp assay
Mm.
that can detect mutated nucleic acid. So those clamp are PNA molecules. Those are like complementary to the wild type.
Okay.
So they hybridize strongly with wild type already forms duplex.
Mm.
So so now wild type molecules are already blocked with clamp. We have only mutated nucleic acid that can bind to target molecules. And this the clamp assay we first published in Nature Chemistry in 2015.
Okay.
And we checked that we detected only like free mutated RNAs, not like DNA, because DNAs are already double stranded. So
Right.
so they can like hybridize here. Now the thing is, you can melt DNA by heating them. You can denature them,
Mm.
dehybridize them. But when they cool down, they will hybridize back again,
Yeah.
right? So detections of like double-stranded DNA is not like easy. So
Yeah.
we, excuse me, we expanded our that electrochemical clamp assay to detect double-stranded DNA. We call DNA clutch probe now here.
Mm-hmm.
Dehybridized DNA, and then we use the clutch, PNA clutch that binds strongly with DNA compared to like DNA. So PNA DNA bond is stronger than DNA. So the PNA binds to one of the strand, and one other strand remains like unhybridized.
Okay.
And in addition to that, we use our previously developed clamp to block wild-type DNA, keeping mutated single-stranded DNA unhybridized. Now that can hybridize to the probe molecules.
So
That's how
I
we
have a quick
detected.
question here.
Okay.
Okay, so why would PNA bind stronger than DNA? What's the property why PNA would bind?
Uh, PNA we call DNA mimic.
Right.
So DNA peptide nucleic acid, it has all types of like DNA bases, like
Okay.
HCG, everything is there.
Mm-hmm.
Only in DNA it has phosphate backbone that is negatively charged,
Mm-hmm.
and PNA is neutral. So instead of phosphate backbone, PNA has peptide backbone. But
Mm-hmm.
it has same all the bases, HCG, everything there.
Oh, okay. Mm-hmm.
So now for like DNA, there is still bonding from the like bases, DNA bases, but there's some repulsion from
Mm-hmm.
phosphate to phosphate.
Yeah.
In other case, in PNA, there's no negative charge and no repulsion,
All
only
right.
like DNA
It will,
pairings.
yeah.
So it bonds like strongly with DNA.
Mm-hmm.
Even PNA bonds is even more stronger than DNA
Mm-hmm.
because both are neutral.
Okay. So do we need to hit in this case to kind of dissociate this kind of DNA?
Yeah, so we heat to dehybridize DNA. Then we add like in a mixture DNA clutch probe and PNA clamp, so that can quickly hybridize to the unhybridized or single-stranded DNA,
Mm-hmm.
and clamp binds to like wild-type DNA. So
Okay.
we have like free mutated single-stranded DNA.
Okay. Got it. So that's so wonderful. And
Yep.
let's move on to your current projects. So now you have your own group and you have started career as an associate professor in Department of Chemistry. So what are some of your current projects? What are you working on nowadays?
We currently focusing on developing wearable sensors that we already discussed. So the sensor can detect like target molecules continuously with minimal invasive way. That is like pretty similar to like continuous glucose monitor. But
Mm-hmm.
instead of like detecting glucose or small molecule, we are targeting to detect real protein biomarkers that are related to different types of diseases. So we are focusing on heart failure, inflammation, uh,
Mm-hmm.
and different other diseases.
Okay. Okay.
So we're targeting heart failure. Just for example, if you think um, we have now smartwatches that can tell us like blood pressure or like
Oxygen level.
yeah, yeah. So those are like physical marker. We can
Mm-hmm.
get something from that, but they cannot like detect real protein biomarkers. They don't like use any types of markers. They're just
Mm-hmm.
physical force. So these are very important. Nice, like wearable detector, but they cannot detect like real protein biomarkers or DNA biomarkers. Those are like even more important. So if like sensor can tell like someone that your that biomarkers or your heart condition looks like getting worse, so you can directly go to doctor to like check out, right? So that can like save a lot of people's life from heart failure or some other diseases.
Okay, okay. Thanks a lot, Dada, for sharing all this research in simple terms for us. And you have so many other wonderful publications and research projects. So I'll share your Google Scholar link in the description section. So whoever is interested, they can go ahead and read other papers as well. So we'll end our research discussion now, and in the next section we'll talk about career.